Hello everyone. So we are going to start a new chapter, that is your amino acid metabolism. So in this particular chapter, the contents has been divided into three parts. So we are going to start the first part. So what is the basics of amino acid metabolism? So how this metabolism of amino acid, it controls the different process of our body. What are the generation, or what are the mechanism of generation of energy in our body from this amino acids, and how the balance of this amino acid, you can say the nitrogen balance, it is maintained in our body. So we are going to compile all of this stuff togetherly in this presentation. So we are studying the first presentation. Here we are going to have a basic details on how this protein they get metabolized, because you all know that the amino acid, they are the basic building blocks of the proteins, and once these proteins they get degraded, finally you get the amino acid. And this amino acid, how they are going to act on the different parts or on the different mechanism of the human body. So without wasting any time, so let's start the presentation. So with a basic background, first of all, say this protein, we know that these proteins, they are the most abundant, you can say, organic compound which are found in the human body, right? These proteins, they perform different function in the human body, right? Now it has been found that the major part of the body dry weight, it is around 10 to 12 kg, and this major part of the body dry weight, 10 to 12 kg, it is made up of the protein itself, right? Thus you can say the body, it is constituted basically by the proteins with a dry weight of 10 to 12 kg. Now what are their functions? What are the functions of the proteins which are there in your body? So basically they have two functions. One, you can say the structural one, or you can say the static one. Another one, it is the dynamic function. So if you see in the static function, basically you have the structural functions, so maintaining the structural integrity of the body, whereas in the dynamic functions you have the functions of the enzymes. We all know that the enzymes and the hormones, even the receptors for these hormones, they are basically made up of the proteins, right? Now we have found that the half of the body protein, it is collagen. That is collagen, it is a static, or you can say it's a structural function. It is present in the supportive tissue. Now you can say, or you know, that this collagen, they are generally found in the skeletal as well as in the connective tissue of the body, while the other half, it is the intracellular protein. Intracellular protein means the proteins which are generally found within the cells. Now you see, these proteins, we all know that they are the organic compounds which contain a nitrogen. Thus you can say the proteins are the nitrogen containing macromolecules in the body, right? Now this protein, they consist of the L amino acids repeating units, and these amino acids, upon the repetitions, they finally form the proteins. The next thing, it is that out of these 20 amino acid, half can be synthesized in our body, right? And these amino acids, they are now divided into two parts. These are the essential as well as the non-essential amino acid. The protein on degradation, they release the amino acid. Sorry, the protein on degradation, they release the amino acid, and each amino acid, they undergoes its own metabolism in the body. Thus you can say the protein metabolism is more appropriately learned as a metabolism of amino acid, right? Now what are the amino acids which are involved? So basically you should know that an adult person, they have around 100 gram of free amino acid, and this 100 gram of free amino acid in the body, it represents the amino acid pool of the body. The pool, you can say, it's a pond from where the, as in a pond, you can see in a pond the water are there, right? So from the pond you are going to use the water. So similarly here, from the amino acid pool, the body, it is going to utilize the amino acid. So basically you will have at least 100 gram of free amino acid in the amino acid pool. Now in this 100 gram, here you should know that the glutamate and glutamine, these constitute around 50% in the amino acid pool, and essential amino acid EAA, it constitutes around 10% of the body pool. Now the concentration of the intracellular amino acid is always higher than that of the extracellular amino acid. Why it is? It's a big question. Why the concentration of intracellular amino acid is higher? Because the intracellular amino acid, they perform different function in the human body, which includes starting from the enzymes, hormones, their receptors and all, right? Now this amino acid, they enters as they have high concentration, right? So they enters the cell against active transport, that is against, you can say, against concentration gradient. The amino acid pool which is there in your body, it is generally maintained by the two basic processes. It is known as simple, the input and the output. So what is the input and the output? It is nothing but, you see, input, it is the source of the amino acid you are inputting in your body, right? And the metabolic pathway that utilizes the amino acid, it is the output. The next thing is the source of the amino acid pool. So from where the amino acids are coming into the pool. So basically, first of all, turnover of body protein. This is the first source. We are going in the details in the next slide. What is turnover? So first of all, you should know that the turnover of body protein, it is the first source for this amino acid pool. The next source, it is the intake of dietary protein. Whatever the proteins you are taking in your diet, they are going into the amino acid pools. The next source, it is the synthesis of the non-essential amino acid in the body itself, right? Now you should know that I'm again repeating the same thing, that about 100 gram of free amino acid, it represents the amino acid pool of the body. Here also I am giving the same line because you should know the quantity. Now you see the concentration of the intracellular amino acid, it is always higher than that of the extracellular amino acid. We have already discussed, right? Now you should know that these amino acid, they enters the cell against a concentration gradient, right? And the turnover of the body intake of the dietary protein and synthesis of the non-essential amino acid, it contributes to the amino acid pool. So these are some of the important things regarding the proteins which are found in your body and the amino acid pool. Now what is protein turnover? As we have already discussed protein turnover. So what is protein turnover? So basically you should know that the proteins in the body is in a dynamic state. The proteins in your body, they are in a dynamic state, means their concentration or their amount, it changes with time, right? Now about 300 to 400 grams of protein per day. Just remember the amount. About 300 to 400 grams of protein per day is constantly degraded. It is constantly degraded and synthesized, which represent the body protein turnover. I repeat, the amount, it is the 300 to 400 amount of the protein. It is constantly degraded, and as I already told you, it is in a dynamic state. It is degraded and again synthesized, and this thing actually represents the protein turnover in the human body. Now control of protein turnover. So which part it is going to control the protein turnover? The protein turnover, it is actually influenced by many factors of the human body, but the basic and the most important thing is that there is a small polypeptide. It is also known as the ubiquitin, with a molecular weight of 8500. This ubiquitin actually tags with the protein and facilitates their degradation. Thus you can say the ubiquitin, it is responsible for the degradation of the proteins, right? Moreover, certain proteins with amino acid sequence like proline, glutamine, serine and threonine, they are rapidly degraded in the human body. I repeat, the proline, glutamine, serine and threonine, these are those classes of the amino acids which get easily degraded in the human body. Now dietary protein. So whatever the protein you are taking in your diet, what is the fate of that protein? Where these proteins they are going? So basically you see there is always a regular loss of nitrogen from the body due to degradation of amino acid. We know that because we used to take the proteins regularly, and these proteins, they are going out from the body. As we all know that these proteins, they are made up of the amino acid, and this amino acid is nothing but it is basically made up of the nitrogen. Thus you can say that there is always a constant amount of the loss of nitrogen from the body, right? Now about 30 to 40 gram of protein, it is lost every day from the human body. It is lost every day. 30 to 50 gram of protein, it is lost every day from the human body, and this amount of the protein, it is supplied through the diet to maintain the nitrogen balance in the human body, right? Now there is no storage form of amino acid in the body. You can see that this glucose, when they are in excess in the human body, they get stored in the form of the glycogen. But if the amount of the protein, or you can say if the amount of the amino acid increases in your body, these don't get converted into any storage form. Now excess intake of the amino acid, whenever you take excess of the amino acid, it is oxidized to provide the energy for the body. So you can see, or you have the examples, right? If somebody of you might sometimes have joined the gymnasium, so during that time, or you can do any type of gymnastic, or you can say physical exercise or running, or any types of athletes, at that time the coach or your trainer, they generally suggest you to take high amount of the protein, because this excess of the amino acid, whatever you used to take, that is oxidized in your body to provide you the energy, the required amount of the energy, right? Now the next thing is that utilization of the amino acid from the pool. So how these amino acids, they are going to utilize, or what are the functions? So basically these proteins, they functions as the enzymes, hormones, immunoproteins, and contractile protein, etc. in your body. So from where these proteins are coming? These are coming nothing but they are coming from the amino acid pool from your body, right? The next one is that the most important nitrogenous compounds in your body, like porphyrins, purines, pyrimidines, they are generally produced from the amino acids, and about 10 to 15% of the body energy requirements, whatever the body it is utilizing the energy, or it is using the ATP, that is derived from the amino acid only. And this amino acid, they are converted into carbohydrates and fats, but as such they don't have any storage form in your body. Now let's discuss the protein digestion. So how this protein, once you are going to take this protein in your diet, how these proteins, they are going in your system, how these proteins are digested in your system. So I am going slowly to have a brief discussion on protein digestion. So first of all, you see digestion in the stomach. So how do this protein, they get digested in your stomach? So basically you should know that the digestion of the protein, it is stimulated by the food acetylcholine. Whatever the acetylcholine is present in your food, apart from the acetylcholine, the histamine and the gastrin, these are released into the stomach. Once you are going to take the protein, immediately it is stimulated by the food acetylcholine. The amount of the acetylcholine which is present in your food, it is going to stimulate it, right? And the histamine and gastrin, these are released into the cells of the stomachs, right? Now the combination of these three, acetylcholine, histamine and gastrin, they stimulate, these three, they will stimulate the stomach to release the gastric juice. Clear? Now mucin, you know that mucin, what is the mucin? It is always secreted in the stomach. This mucin, they have a protective, they made a protective layer over the surface of the stomach, right? And HCl, this HCl, they are responsible for maintaining a pH of the stomach. The pH, the optimum pH of the stomach, it ranges from 0.8 to 2.5. It is the pH range of the stomach, right? And this HCl, it is secreted by the parietal cells, which are generally found in the stomach. And the next part, it is the pepsinogen. So this pepsinogen, it is secreted by the chief cells, and this pepsinogen, as we all know that it is a zymogen. This pepsinogen, it is responsible for the majority of the protein digestion, or you can say degradation in the human body. Now the proteolytic enzymes and their activation. How do these enzymes they get activated? So first of all, you see three enzymes are in the gastric juices. What are the three enzymes? Basically, you have the pepsin, you have the gastricsin, and you have the renin. These three enzymes, they are in the gastric fluid. I repeat, pepsin, gastricins, and renin. All these enzymes, they cleave the proteins into the peptides, right? Now we know that the pepsinogen, it's the important zymogen. Now it is activated, this pepsinogen, it is activated by the pepsin enzyme itself, right? And which is already present in the stomach. And apart from this pepsin, the pepsinogen, it is also activated by the HCl, which is secreted by the parietal cells in the stomach. The next part, it is the this pepsinogen, actually it is cleaved off. This pepsinogen, it is a zymogen, it is cleaved off by the enzyme pepsin itself, right, to form the enzyme pepsin and a peptide fragment. And this peptide fragment, it is further get degraded. It is of no use in the human body. Now this pepsin partially digests the proteins by cleaving the peptide bonds, which is formed by the aromatic amino acid like PHA. It is the phenylalanine, tryptophan, and tyrosine. So phenylalanine, tyrosine, and tryptophan, these three are aromatic amino acid, right? So this pepsin, actually it is going to digest the protein by cleaving the peptide bond, the peptide bond which is formed by the aromatic amino acid. These three are the aromatic amino acid in your body, right? Now the optimal pH for the gastricsin, it is 2 to 3. It is the range. So the ratio between the gastricsin and pepsin in the gastric juice, it is always 1 is to 5.5. This is the ratio. So the ratio between the gastricsin, it is the amount of the gastricsin, it is less, that is 1, and the pepsin, it is 5.5. This is the ratio, and the pH should be 2 to 3 for its optimal function. Moreover, this ratio can be changed, which based on the pathological condition, like different sort of diseases, right? Now regarding the renin, this renin also, they possess a proteolytic activity and causes the rapid coagulation of the ingested casein. Casein is a protein, you know, it is generally found in the milk products. So that this renin, they also possess a proteolytic activity and they causes the rapid coagulation of the ingested casein, whatever you used to take, right? But this enzyme plays an important role in the children because the optimal pH for it is 5 to 6. You can see here the pH, it is 5 to 6, but in the adults the range of the pH, it is 0.8 to 2.5, right? Now digestion in the duodenum. So what is the role of the duodenum and how this protein they get digested in the duodenum? So first of all, you should see, or you should know that the stimulation for by the food secretin and the cholecystokinin, these regulate the secretion of bicarbonate. This bicarbonate, it plays an important role in the duodenum. So stimulated by the food secretin and the cholecystokinin regulate the secretion of bicarbonate, zymogen, trypsinogen, chymotrypsinogen, and proelastase and procarboxypeptidase by the pancreas into the duodenum. So regarding this thing, I am going to details in the next slide. So bicarbonate, what actually do this bicarbonate, they changes the pH in the duodenum, they changes the pH to 7, right? So you see here the enteropeptidase, so which is secreted by the mucosa of the duodenum, it initiates the activation of the pancreatic proenzymes. So how it is going to stimulate, you see in this figure. So this is the enteropeptidase, right? This enteropeptidase can convert the zymogen trypsinogen into trypsin, right? Now this trypsin, it can also convert, first of all we are going to convert this chymotrypsinogen into chymotrypsin. Again it can convert the proelastase into elastase. Even it can convert the procarboxypeptidase into carboxypeptidase. Further it can convert the zymogen prolipase into lipase. So basically this is the overall nature of the chemical changes which takes place in your body, right? Now digestive tract of protein. So how the tract it is maintained within the human body. So basically these proteins are generally too large to be absorbed, because we all know that these proteins are made up of amino acid, and these proteins, they have high molecular weight, right? Thus these proteins are too large to be absorbed by the intestine, and therefore this protein must be hydrolyzed to amino acid, or else these proteins cannot be utilized by the body. First, next one is that the proteolytic enzyme responsible for the hydrolysis are produced by three different organs. So what are the organs which are synthesizing or which are contributing to the digestion of the protein? These are the stomach, the pancreas, and the small intestine, right? The small intestine, it acts as a major organ for the digestion of the protein into the amino acid. Now in the stomach, we have already discussed. So in the stomach, basically the HCl, it is secreted by the parietal cell, and the pepsinogen, it is secreted by the Schiff cells, right? Now the pH of the gastric juice, it is around 1. Now the food, it is retained in the stomach for 2 to 4 hours. So this is also known as the gastric retention time. So basically we considered it as a 2 hour, but based on the physiology of different people, it is diverted, or you can say it have a range of 2 to 4 hours, right? Now what is the function of the HCl? This HCl secreted by the stomach, it actually kills the microorganism. Moreover, this HCl, we all know that this is a strong acid, so it denatures the protein and provides the acid environment for the action of pepsin. Because when we suppose the pepsin to act on a protein, the first and primary criteria is that it should have a acidic environment. Moreover, autocatalysis. Autocatalysis means the pepsinogen, it is converted to active pepsin by the HCl itself, right? Now what is the contribution from the pancreas and a small intestine? So basically endopeptidase, it is secreted by the pancreas, right? So trypsin, it will go and act on the carbonyl of arginine and the lysine, right? Chymotrypsin, it is going to act on the carbonyl of the tryptophan, tyrosine, phenylalanine, methionine, and leucine. These are the amino acids where this chymotrypsin, it is going to act, right? Moreover, elastase, this is going to act on the carbonyl of alanine, glycine, and the serine. Next is the exopeptidase. This exopeptidase, it is secreted by the pancreas itself, right? Now this exopeptidase, it have the carboxypeptidase, what I have shown in the previous slide. So this carboxypeptidase, carboxypeptidase A, they actually act on the amine side chain of the alanine, isoleucine, leucine, and valine, right? Moreover, the carboxypeptidase B, it acts on the amine side chain of the arginine and the lysine. And the aminopeptidase, it is secreted from the small intestine. It cleaves the N-terminal residue of the oligopeptidase, or you can say oligopeptidase, right? Now what is the nitrogen balance? We know that the protein, they have the nitrogen in them, right? Now this protein, you are taking them in your body. Now this nitrogen balance, it is calculated based on the amount of the nitrogen input as well as the amount of the nitrogen output. So basically you see here the nitrogen balance, it is a comparison between the intake. You can see here the intake, that is the nitrogen intake, and the nitrogen loss, right? So how nitrogen it is lost? So nitrogen, the loss can be regarded as the loss due to the undigested protein in your fecal matter. Moreover, non-protein nitrogen, sorry, this should be nitrogen, not nitrogen, non-protein nitrogen such as the urea, amino acid, creatinine, uric acid in the urine, sweat, saliva, and the losses by hair, nail, and the skin. Now nitrogen balance, it is important in defining the overall protein metabolism of an individual. Moreover, it is also important for defining the nutritional nitrogen requirement. So what are the scale of the nitrogen balance? So nitrogen balance, basically it have three scale. Either it will be positive, or it will be negative, or it will be neutral. So what is a positive nitrogen balance? It is nothing but if the amount of the nitrogen entered in the organism is more than the amount of the nitrogen removed from the organism. If the input is more and the output is less, then this is a positive nitrogen balance. So it generally found in the young growing organism. Moreover, during the recovering after severe disease and using anabolic medicine during the time of pregnancy or lactation, or you can say convalescence, right? The next type, it is the negative type. Here the loss, it is more as compared to the input. Thus the amount of the nitrogen removed from the organism is more than the amount of the nitrogen entered in the organism. So it occurs during the time of senile age, you can say in the older age, or it also happens during the time of destroying of the malignant tumor, or you can say vast combustion or poisoning by some toxin. So high loss of the tissue protein in wasting disease like burns, hemorrhage, kidney disease. In this condition, actually negative nitrogen balance, or you can say negative nitrogenous balance, is seen in a body. And what is the third scale? It is the neutral one. So zero nitrogenous balance. Here input is equal to the output. So it generally occurs in the healthy adult people. So normal adult, they will have a nitrogen equilibrium, that is the loss, it is equal to the input, or you can say intake. Now regarding the protein turnover. So first of all, protein turnover. What is protein turnover? It is nothing but the degradation and a resynthesis of the protein. That is the amount of the protein which is degraded and the amount of the protein resynthesized in a body. It depicts the protein turnover in the human body. So half lives of the protein, it varies from minutes to years, right? Moreover, the structural proteins, usually they are stable. You can say, for example, the protein crystalline, which is found in the lens, it is stable throughout the lifetime of an organism, right? Regulatory proteins are generally short-lived. So what are the regulatory proteins? These are the enzymes, the hormones, and their receptors. These are generally the short-lived protein. Now we know that what is ubiquitin. Ubiquitin, it is nothing but it's a tag. Ubiquitin, it marks the protein for destruction. So once the protein, it is tagged with ubiquitin, the fate of the protein is finally degradation. Means the protein is supposed to be degraded in the body. Now you should know that the ubiquitin is a small protein which is found in all the eukaryotic cells. Moreover, it have a structure, that is the structure of the ubiquitin is nothing but it have a extended carboxyl terminus. It have a carboxyl terminus of the glycine that is linked to other protein, and this glycine residue from the linking additional ubiquitin molecule. Means say, you can say the ubiquitin, it is made up of the last chain of the ubiquitin, it is the glycine, right? It is linked to other protein, and the lysine residue of this ubiquitin, it links the one ubiquitin molecule to another ubiquitin molecule. So this is the overall chart. So here you can see the diet protein. Now it is contributing to the amino acid pools. From here it is this from this amino acid pools, the amino acid are used for tissue proteins. You can say they are used for non-protein nitrogen derivatives, right? And sometimes transamination takes place where the amino nitrogen in the glutamate, it led to the deamination. Finally it is coming out from the body as a urea. Even acetyl coenzyme 8 will act here, it will enter the citric acid cycle and it is coming out as a carbon dioxide. These are the overview of the protein metabolism, regarding which we are going to discuss in the part 2 and the part 3 of this amino acid metabolism. So lastly, thank you everyone for your patience hearing. If you like the video, press the like icon. You can share this video with your classmates and colleagues, and for further notification from this channel, you can press the bell icon. So thank you everyone once again.